Hey everyone. Okay, that is possibly the quickest I have ever got this all set up and ready. So we are going through math skills for A-level biology paper 1. What I would say is, just while we are letting everyone get the notification, sign in. I say sign in, basically just watch. You could, while you're waiting for us to properly get started, head to this link. If you want this document for free, it's missestrick.co.uk/free-maths. And what you get is this entire 12-page booklet, which has got the list of all the math skills which are specific to paper 1 and also paper 3, but paper 1, and then also the exam questions that we're going to do. But it's not just a mark scheme, because what I hate about the mark schemes is that they don't actually tell you how they got that answer. They just give you the answer. So I've got the workings for all of the questions as well. So if you do want this, it is completely free. You just need to download it from missestrick.co.uk/maths. So that is completely for free if you want to get your hands on this, so you don't have to try and make notes as you're going through. So what I'm going to do is just show you the booklet so you can see what are the math skills that are likely, well, not likely, but they could come up on paper 1, because that's what it says on the specification. So on the spec, what I've done is picked out the math skills linked to topics 1, 2, 3 and 4 in the spec where it says MS skill, so math skill. Now, just to point out, that doesn't mean this is the only thing that could come up, because you can still get generic maths questions, like anything to do with algebra or percentage change, those sorts of things. But this is particular math skills linked to the topics for paper 1. So let's jump in then. That was what I was just reading out, basically what is this booklet going through. So it's the math skills from the spec linked to the different topics. So starting with topic 1, in the enzymes topic, it does actually say calculate the pH from hydrogen ion concentration using this formula. I have never seen this come up, and this will be, I was going to say the 10th year, but it changed in 2015. The first exam was in 2017. So 2017, 18, 19, 20, 21, 22, 23, 24. We've had 8 years worth of papers, and I've never seen this come up. I mean, I suppose you could justify it's come up in a few ways where they've told you something that you could rearrange. But anyway, it's there. Next, plot data from the enzyme practical in an appropriate graph format. So that's basically plotting data to create a graph. Using a tangent, we're doing one of those questions tonight. Using a tangent to calculate the initial rate of reaction. No idea what that was. My phone just started making noises on TikTok. Must be something strange I don't know about. Okay, then that's what you can get for enzymes. And this one here links to the required practicals as well. DNA structure, we're going to be doing one of these maths questions tonight as well. So using base frequencies to calculate the proportion of other bases in complementary DNA strands. So they explicitly point that out as a math skill, meaning that could come up on paper 1, but also paper 3, but we're just focused on paper 1 for today. Then we get to topic 2, studying cells, the magnification calculation. So magnification is image size divided by actual size. So you could have that one. Let's turn this over. Cell cycle. So here you could work out, or be asked to work out, the mitotic index. The formula is here for you. So the number of visible cells in mitosis divided by the total number of cells. Or using a scale bar to calculate the actual size of cells. And we are doing that as an exam question this evening. That's one of the ones we're going to have a go at. Transport across membranes, so plot results for permeability or osmosis experiments, determining water potential from the intercept of a graph. So that's where you might have your calibration curve. You then have an unknown, and you can read off from the graph to work out what your unknown water potential is. Or in the case of water potentials, it might be to work out the concentration inside of the plant tissue if you've been doing the osmosis required practical. Topic 3, jumping onto this one then, is calculating the surface area to volume ratio. And this where it links to in the math skills, it says that you need to know how to calculate the area or volume of different standard shapes. So for example, you need to know for a circle, the area is pi r squared. I have to say, although it says in the spec you need to know the standard volumes, they've never actually asked for volume. They've always given it to you. Right, I'm going to have to do a quick 30-second break or so because one of my children's calling. I'll be right back. Okay, and I am back. I did actually see as I was coming back, someone was asking for the link. I've put it at the top of every page. So if you do join partway through, you can see. So it's missestrick.co.uk/free-maths. This booklet is completely free. So you don't have to manically try and write notes. You can just download it for free, use it for your revision, as well as all the questions and mark schemes we're going to do. Okay, so then gas exchange topic 3 is working out, it's cut off a bit there actually, but the pulmonary ventilation rate, so tidal volume times your breathing rate, or you could be asked to rearrange that to then work out the tidal volume or breathing rate. 3.4 genetic diversity, so this is actually topic 4 now. Oh no, 3.4. I don't think I've been saying the right topics here because these are actually just the math skills or the list, because anyway, this is topic 4, so I don't know why it's going on to 6 and 7 there, but those might be like the subsections, can't remember, but the point is it could still come up. This one here is something that we're going to be using, and as I keep saying, meiosis hasn't come up in a while, so it seems quite likely meiosis could come up. This is one of the math skills linked to meiosis, knowing that if you're starting with one cell and it divides, let's say 30 times, how many cells would you have at the end? It's 2 to the power of 30, 2 to the power of n, to work out how many cells you'd have at the end. So we're going to do a question linked to that so you can see how that's used. Log scales. So you use log scales when you have a really big range in your data, meaning your y-axis, you might have to try and go from like 10 all the way up to 1 million or 1 billion, and you're not going to be able to pick a scale that goes through that range and still shows you the pattern on the graph clearly. So that's when you would then convert your raw data into log data and you'd get your log graph. There's also reading off the log scales. If you actually have log graph paper, then every line means like 1 times 10 to the 3, 2 times 10 to the 3. That probably isn't particularly clear right now, but I do actually have a TikTok and an Instagram post on it. I'm not sure the easiest way for you to find that though, because it was posted quite a while ago, but I do have one going through those concepts. Right, then we've got index of diversity. We're going to be having a go at that one this evening, because the most common thing that people do wrong here is the denominator, not knowing what this lowercase n actually stands for. And it's such a common maths question that comes up, so I want to make sure everyone is confident on this before their exam next week. Right, then, carrying on with that, calculating mean values, that's pretty straightforward. Interpreting standard deviation, you are not going to be asked to calculate standard deviation. You wouldn't be asked to calculate student t-test, correlation coefficient, or chi-squared. They can't ask you to do any of the statistics because that would end up being too long, too many marks on maths in a biology paper. They could give you, for chi-squared before, they have given a table to guide you through calculating it, but when they did that, it was things like observed minus expected, so it's just subtraction. But they won't ask you to calculate the entire stat, it's just interpreting what that means. So that's this document, that is it. I say it, I know that was 3 pages of math skills, but that's the first part of this document. It's just going through what I've pulled out from the spec, every topic specific math skill that could come up on paper 1 and then also paper 3. And from that, I've picked out some of those for us to have a go at as exam questions. And as I keep saying, you can download this completely for free, missstroke.co.uk/free-maths. So if you want to have this booklet, so you've got the list and these exam questions and the mark scheme, it's there for you for free to help you before that exam. Okay, question 1, let's have a go. And do you know what I realised? I was getting everything set up for this session. I was like, yeah, good, got my different cameras all aligned, it's all good to go. And then I realised I don't have a ruler. So every question that needs a ruler, I'm going to be having to use a tape measure, which is going to significantly impact my accuracy here. So there's a little lesson to everyone. Make sure you've got a ruler for your exam on Thursday, because you're probably going to need it at some point, just like I will this evening, and I'm not going to be able to use one. So let's jump into the questions. Amylase is a digestive enzyme that hydrolyzes starch. So amylase is digestive enzyme that hydrolyzes starch into maltose. Students investigated the hydrolysis of starch using amylase by recording the concentration of the product maltose that was produced at set intervals. So we've got concentration of maltose in milligrams per decimeter cubed, and then the time. The first part then is we're asked to use a tangent to determine the rate of reaction at 15 minutes. Now on this one, they've actually said use a tangent. Sometimes they don't explicitly say use a tangent. So the way that you know you need to use one is, if ever you're asked to work out the rate of the reaction on a point on the graph where it is curved, you need to use a tangent. If you've got a straight line of best fit, you don't need to draw a tangent, you've already got a straight line to work out the gradient from. So if you've got a curve, you have to draw a straight line, which is our tangent, and then work out the gradient of the tangent to get our rate of reaction, because our rate of reaction is going to be our concentration divided by time. And if you weren't sure on that, then you just look at the units. We can see we've got milligrams per decimeter cubed per minute, which means divided by time. So that's how you know it's divided by time. So at 15 minutes, here we go. We need to get it there. Oh, this is going to be awful. Let me attempt to draw a tangent with a soft tape measure. Please don't replicate this. This is embarrassing. But it was the best thing I could find. And it's not even see-through, so I can't check I've done it in the right place. Okay, so for your tangent, the main thing that you need to have is, first of all, it needs to be touching at exactly that point. Then the angle between your tangent and the curve, either side of where it touches, needs to be equal angles. It doesn't matter how long you draw your tangent because it's a straight line. So wherever you work out the gradient, it's always going to be the same because it's a straight line. So I've done mine from here, which looks like it's partway through there. So 250, 60, 70. Let me just check. So those are going up in tens. So that would be 200. And the top part of it is 295. And that is 240. So I'm now working out the difference in my y-axis and then divide that by the difference in this bit here between the x, which I've done a bit easier because that is 20 minus 10. So let's go 295 minus 240. So we've got 55 divided by 10, 5.5. So that's what I'm getting as my answer, 5.5. Now, if you do have a go at these questions and print it out and you don't get exactly 5.5, do not panic, because in these tangent questions, there's always a range of answers they'll accept, because they realise people's tangents aren't going to be exactly the same. So they give you a range that they'd accept to take into account the slight different positions that you might put your tangent in, which it would still be acceptable. Right, so that's the first maths question. Next then, we've actually got some of the theory questions in there as well, but it's all good revision. Explain why the graph plateaus. So we've got the concentration of maltose, which is our product. The amount of product being produced, it plateaus over time. The reason for this is, I think I'm running out of ink here. Let me see, I might need to change pens. Oh yeah, takes me back to being at school, that running out of ink. Oh, it's a shame. I always try to use that pen now because my handwriting's so much nicer in fountain pen. And when I do it in ballpoint pen, it's awful. And people always tell me in the comments how awful my handwriting is. But we'll have to deal with it. So explain why the graph is a curve that plateaus. That's what we're saying. So concentration of maltose, that's the product from starch. So the reason it's going to plateau is starch, or in fact, I'm going to say all the starch. All the starch is now digested into maltose. So no more maltose is produced. So that's what I'd be saying. That's basically the reaction's finished. So having a look at our answers, we've got, that's just talking you through how to do it. Because like I said, you get the answers of how to actually do this. So draw your tangents, the difference in y divided by the difference in x, and you've got the units there. So your rate of increase in concentration of maltose slows as the substrate is used up. The curve plateaus because all the starch is used up. So that was our answers for that one. Next question, we've got DNA is a polymer made up of nucleotides bonded together by phosphodiester bonds. Describe how these bonds form between the nucleotides. Okay, first thing I'm going to say is it's a condensation reaction. Right, you have a go at that. My children are clearly quite unhappy by the fact I'm doing maths tonight because they keep calling me. Let me go grab them again. There we go. Right, so condensation reaction. DNA is a polymer, just have to remind myself now, made up of nucleotides bonded together by phosphodiester bonds. Describe how these bonds form between the nucleotides. So I'm going to say a condensation reaction. I'm going to say between the phosphate group and deoxyribose of two different nucleotides. So that's what I'd say for that one. Let's just double check before we do the maths question and forget about it. There we go. So yeah, condensation, or you could say loss of water, and between the phosphate and deoxyribose, which I did. Another mark you could have said if you were wondering, you could have also had as another option using the enzyme DNA polymerase. Okay, just as a reminder, so I can see people keep joining and they're asking, this is a free booklet. It's missesther.co.uk/free-maths. Right, the maths question. This was one of the skills that we saw. Let me just check everyone can see this okay. I think so. Yes, the recording is going to go on my YouTube channel and on my Instagram channel. So there is the recording of this as well. Complete the table to show the percentage of DNA bases you would expect. This is looking at if you had complementary strands of DNA in your double helix. I mean, I drew it. I should just show it. I've got it here. Your two strands, you would always expect to have the same proportion of adenine as you do thymine because they're complementary bases. So on one strand, wherever you have an adenine, it'd be opposite of thymine. So you'd have the same proportion. Same if you're doing the adenine to thymine here, cytosine to guanine, guanine to cytosine. And because it's percentage, each row must add up to 100 because it's 100%. So I could actually work that one out straight away because we just need to make sure it adds up to 100. So 23, 20, this is embarrassing, I'm doing everything on the calculator, but it's just because I'm talking at the same time. 100 minus 81 is 19. Now, if we've got 19 guanine, you would have the same proportion of cytosine, so that would be 19. And if we've got 20 thymine, we would have 20 adenine. So that's how you do these questions. It's the concept that in a double-stranded molecule of DNA, you will always have the same proportion of adenine to thymine and vice versa on these ones. And each strand must add up to 100 because it's percentage. This has come up before where they've shown you viral DNA as an extra row. And they might have said this is a percentage. Oh, let's do some easy numbers so I know it definitely adds up to 100. Up to 50. Okay, let's just do that. But what we can see here is we don't have the same proportion of adenine and thymine and cytosine and guanine. And the reason for that is viral DNA is single-stranded. So you don't have the complementary base pairs, so you're not going to have the same proportions. And that's what they are always after. If you don't have the same proportions of your complementary bases, then it means you've got a single strand of DNA. The only reason you have the same proportions is because it's double stranding, so you have complementary base pairs aligning. Right, let's go on to the next one. And as I said, you've got the workings here for everything that I said, so you can download it or watch the recording, your choice. Next question. Oh, again, I need a ruler, so I'm going to have to get my embarrassing tape measure out, so I can't find a ruler. Below is a diagram of mitochondria drawn from a microscope with a magnification of times 25,000. Use the diagram to calculate the actual length of this mitochondria in micrometers. Okay, so first thing I'm going to do is that's actually the way that I always remember. I know in the spec it said magnification first, but I just find it easier to say I am. Then I can rearrange the formula, what we're working out the actual. So we want to know the actual. So that is going to be image divided by how many times it's been magnified. And we know that the magnification is 25,000. We need to work out the image size. So out comes my highly inaccurate tape measure. If we're talking uncertainty, there's going to be a lot of uncertainty in this measurement. Right, this takes us to 66. I'm going to say 66 millimeters. However, they want the answer in micrometers. So 66 millimeters, we need to times that by 1,000, which means we have 66,000 micrometers. So I actually need to do 66,000 divided by 25,000, or basically 66 divided by 25. So we have 2.64 micrometers. So that's what we do for that one there. Okay, next question. Question 4. Oh, here comes the index of diversity. So we've got ecologists investigated changes in bee communities on a farm. They used data from a number of sites to determine the change in mean species richness and the change in the mean index of diversity. We've got the species, number of individuals. We need to calculate our index of diversity. They often actually tell you that N equals the total number of individuals, which is basically the sum of that. And lowercase n equals total in each species. So let's work out n first. We've got 3 plus 19 plus 3 plus 3. So we've got, let's do it in my head this time. 9 plus 19, 28. So capital N equals 28. So that means we need to do D equals 28 times 28 minus 1, which is 27. Then we need to do our lowercase n. So that would be 3 times 2 equals 6. That's the same for all of these. And 19 times 18. The reason I'm doing that is it's n times n minus 1. So 3 times 3 minus 1. And it's the sum of all of those. So we've got 18 times 19 equals 342 plus 6 plus 6. Hang on. 3, 4, 2 plus 6 plus 6 plus 6. So that means the sum of that is 360. So 28 times 27. I do recommend you do actually work that out, because if you get the final answer wrong, whether you're just wrong or maybe you've typed it in wrong in your calculator, they usually give you, if you get it all wrong, you usually get 1 mark for getting either the denominator or the numerator correct. So that's why I'm writing it literally out. Even though I know you can do it all on your calculator, if you make a mistake, you can at least pick up marks as you go. So I do recommend you write out each stage as you go. So 756 divided by 360 equals 2.1. So 2.1. And just to give you a bit of context, the index of diversity using this formula typically falls between 1 and 10. It doesn't have to be that, but most of the time it falls between 1 and 10. So if you've got a number in the negatives, it's obviously wrong. If you've got a number that is much, much bigger than 10, then you've gone wrong somewhere. Test it, try again. So between 1 and 10 is the most likely outcome. And as I said, just in case you aren't sure how to do this, if you do download this free pack from missestrick.co.uk/free maths, you get the questions, you also get the mark scheme, but I've written out the steps for how you actually calculate it as well, in case when you come to do it yourself, you just can't remember. So there you go, you've got the calculations there. Next question. In an investigation of growing bacteria, there was a population size of 1,340 bacteria. Bacteria divide every 20 minutes. Bacteria were left to divide by binary fission for 5 hours. Assuming no bacteria died, what's the population size after 5 hours? This is where we need to use the formula 2 to the power of n, where we use 2 as the base number because when bacteria divide, they split in half to create 2. So that's why it's 2. And n here equals number of divisions. So every time it splits in 2, we need to multiply that by the number of, or 2 to the power of, not multiply, 2 to the power of the number of those divisions. So we don't know how many divisions there are, but we do know that they divide every 20 minutes and they're left to divide for 5 hours. So within 1 hour, that's 3 divisions, but we've got 5 hours. So that means it'd be 3 times 5 equals 15. So we have 15 divisions. So that means I need to do 2 to the power of 15. So let's just do that. 2 to the power of 15. That equals 32,768. However, that would be the answer if we started with 1 bacteria. And we didn't start with 1. We started with 1,340. And all of those are going to create this many cells, because every one of those cells will divide 15 times. Each time it's doubling. So all 1,340 are going to create this many, which is why we actually need to do for this one, it's 2 to the power of 15 times how many you have to start with. So that is, I've still got it in my calculator, times 1,340. So we have an answer of but it hasn't told us to give it in standard form, but I probably would because, well, often it does tell you to do it in standard form because it's another math skill. But if you don't have to, you might want to. So let's see, we've got 1, 2, 3, 4, 5, 6. So I'm going to say 4.4 times 10 to the 6. And that does sound right to me. If I was doing this, I'd be like, does this sound right? And to me, yes, because we know that bacteria divide very rapidly and it started as a big population already, so I'd expect to have a big number at the end. Okay, and as I said, for every single question, you've seen me answer it, so you can watch it back in the video, or you can watch it here. Did I get a different answer this time? Oh no, I just rounded up, that's why. That's fine, I just rounded up. I was just thinking, I was like, what have I done wrong here? Okay, so that is it. That's the end of the pack. I haven't gone through every single math skill because I was only aiming to make it about a half-hour lesson, and it was pretty much spot on half an hour. So that worked very nicely with the timings. So let me just say, there's one final thing, if you've only just joined in now. So that was going through AQA A-Level Biology Paper 1 Math Skills, basically the skills specifically linked to topics 1 to 4. You can get this booklet for free, missesterett.co.uk/free math skills, where it lists all the math skills for you that you could need to know, as well as all the generic ones that aren't listed here. Then I've got some of those ones, the questions, the answers, with the answers explained. So that's all there for you. If you do want even more help, this one isn't free because it does cover literally the entire A-level with every single math skill explained, modelled, practice questions, answers explained for the entire A-level. But if you do struggle with the maths and it is 10% of your A-level, then you can get the AQA A-Level Math Skills Workbook, which is on my website as well. But you've got this one here for free if you don't want to buy anything to cover some of those math skills. So that is it for this evening. Best of luck, everyone, for your exam next week. And if you're coming to my live lesson the night before the exam, that's my pick and mix lesson on extended response, I will see you then, because that's our hour lesson going through the theory and skills for extended response. But I'm going to leave it there. I think some people are saying you missed off a 0 when you wrote it out. Oh, I might have done because I'm kind of talking at the same time. So I might have missed it out if it was meant to be to the power of 7 and I didn't write down one of those zeros. Okay, so someone did just, I'm going to answer this question. How do you know how many significant figures to leave an answer to if it doesn't specify? Usually, if it doesn't specify, they will accept a range. Sometimes it's you need to match it. By the way, I'm just kind of wrapping this up at the same time. It's a bit therapeutic, so I'll do that as I'm answering. If you've been given a table of data or other numbers and they're all to 1 decimal place, you should give your answer to 1 decimal place to match what you have been given. If they don't, then typically we'd go to maybe 2 or 3 significant figures or 1 decimal place. Right, I'm going to end it there, everyone. Enjoy the rest of your evening and I'll see you in a live very soon.